Now we can summarize our discussion related to cutting of DNA molecules. We have the idea that microorganisms can protect themselves from the invasion of foreign DNA that may be from any other microorganisms or virus particles. तो जब भी कोई फॉरेन डीएनए किसी माइक्रोब के अंदर एंटर होता है तो जो होस्ट का रिस्ट्रिक्शन सिस्टम है उसको अटैक करके तो डिग्रेड कर देता है. सो इन दिस वे microorganisms they can protect themselves. We also have the idea that restriction enzymes they may be a variety of type. लेकिन सारे के सारे enzymes they are not useful in the process of gene manipulation. इसकी रीज़न क्या है? रीज़न ये है कि ज़्यादातर जो रिस्ट्रिक्शन एंजाइम्स हैं वो डीएनए को रैंडमली ब्रेक करते हैं. They can cut the DNA more randomly. But another category which is very well known, and it is type 2 restriction enzymes that are valuable in the process of gene manipulation, and they are mostly used to create artificially recombinant DNA molecule. So we are also aware that type 2 restriction enzymes, when they cut DNA fragments, so different type of fragments they are produced. Now these fragments they may have different type of ends. Like some DNA they have blunt ends. For example, if DNA it is digested with He3 restriction enzyme, the DNA fragments that are produced they are having blunt ends. Blunt end means that there are no unpaired bases at the end of DNA. So both fragments of DNA they have equal length. On the other hand, some fragments of DNA after digestion with restriction enzymes they will have sticky or cohesive ends. I think you are well aware that some fragments they have 5' or 3' unpaired bases, so that are complementary with other DNA unpaired bases. So that's why they are called as cohesive or sticky ends, because at 5' or 3' ends the overhang or protruding of a few base pairs are present that are complementary with other DNAs if they are digested with same restriction enzymes. Now these single-stranded overhangs, as I told you that it may be present at 5' if DNA it is digested with EQR1, BAMH1 or even HIND3. And in some cases, if the DNA it is digested with PST1, these overhangs or sticky ends they are present at 3' ends. So the DNA having blunt end or sticky end depending that which restriction enzyme has been used to digest the DNA. Then you are also well aware with the nomenclature of restriction enzyme that generic name, first letter and two letter of the specific epithet they are used to name the enzymes. Then it is also important that which strain it is there and what is the number of restriction enzyme that has been isolated, because the same strain may produce different type of restriction enzymes. So the enzyme may be 1, 2, 3, 4, 5, and I quoted the example of hint 1, 2, 3 and so on. Now when DNA it is digested with type 2 restriction enzyme, the DNA fragments they may be of different lengths. It will depend that what is the length of a recognition sequence, how many bases are present. Either they are the 4 bases, 6 bases, or the recognition sites may be composed of 8 bases. So if the recognition sequence they are having more bases, the DNA fragments that will be produced, they are having more length as compared to those having low number of nucleotide bases like 4 as compared to 8. It also depends that how much time it is given for the digestion of the source DNA. If more time is given, so enzyme will get more time to digest the DNA, so the length of fragments, it will be less. But if partial digestion is there, partial mean that the less time is given, so the restriction enzyme will not get enough time to attack the source DNA. So the length of fragments that will be produced, it will be high. So it is called as partial digestion. And of course, the frequency of particular recognition sequence, it is also very important. So restriction enzymes, they are very important in the process of gene manipulation.